Okay, I'm going to start talking about Chapter 6. It's actually a subject which is very important in organic chemistry, and I want to spend some time on it. Again, it's not going to be covered on next week's exam. We're just going to cover Chapters 4 and 5. But I don't want to waste too much time getting to this because we are a little bit behind according to my original schedule. So, I want to talk about stereochemistry. In particular, there's an area or a type of stereochemistry which we haven't addressed yet, and that has to do with three-dimensional arrangements of tetrahedral atoms. We've had some examples of stereochemistry already when we talk about ring compounds, right, that are fixed and can't rotate, and you can have groups that are all attached to the same atoms, but they're positioned in space in 3 dimensions in specific orientations, so they could be either cis or trans in terms of a ring, or on a double bond, which is also rigid with that pi bond, and it can't rotate, so you can have groups on the same side or opposite sides of a double bond. So we have cis and trans, or E and Z, depending on what kind of naming system we're looking at. But there's another kind of stereochemistry when we're talking about objects or molecules in 3 dimensions. They can have symmetries associated with them. They can have a lot of symmetry, or they can be asymmetric. And because carbon is a tetrahedral atom, it's possible to have a carbon atom that has 4 different groups attached. And if you look carefully at that, the mirror image of that molecule is something different. Okay, they're not the same. Because again, it's a tetrahedral in 3 dimensions. Same thing, I love this drawing by Escher. Escher, I love all the Escher art because it's interesting, but it's one hand drawing another. Kind of illustrates this point also because in many aspects in nature, there's handedness, and you have a right hand and a left hand, right? You can't superimpose them, they're not the same. You can see that they're mirror images, pretty much, right? Mirror images, but if you try to superimpose them, obviously they're different. Same thing with molecules. And molecules can show this kind of stereochemistry. They are different. The properties of molecules that have this particular kind of stereochemistry there's only a few things about the physical properties which are different. But let's take a close look at this. If we look at a molecule like difluorochloromethane here, and I've shown the tetrahedral structure. This is a carbon atom that has obviously 4 bonds to groups. In this case, it happens that 2 of those groups are identical, the fluorines. 2 fluorines, 1 hydrogen, 1 chlorine. And if you look at the mirror image of that molecule, that's what that mirror plane is supposed to reflect the molecule as the mirror image. Is that molecule different, or is it the same, or what is the relationship between those 2? They're the same. If you think about taking one molecule and sliding it over and superimposing them on top of each other, you'll see that if you slide this molecule over, for example, the chlorine atom will end up exactly the same place as the chlorine atom back there. This fluorine on the left will end up over the same position as the fluorine on the left in the molecule on the left. This fluorine would overlap with that fluorine, this hydrogen would overlap with that hydrogen, that carbon atom would overlap with that carbon. So if you merge these together, you would see it would be exactly the same. This is a molecule which has symmetry. Okay, there's symmetry in this molecule. It happens to have a mirror plane within the molecule bisect. And one of the things we think about when we look at molecules in 3 dimensions is, are there elements of symmetry? Are there rotation axes that are symmetric? Are there planes of symmetry within the molecule itself? And in fact, in this case, if you look carefully at that molecule, if you were to bisect the molecule right in half this way, just think about sticking a mirror plane right in the middle of the molecule. You can see the left half of the molecule would be directly symmetric with the right half of the molecule, right? It's a mirror plane. Now, this happens when you have at least 2 or more of the same group on a carbon atom. That fluorine happens to be the same as that fluorine. But what if one of these fluorines was something else? Okay. Then there's no longer symmetry. And in that case, when we have carbon atoms with 4 different groups attached, and I've just color-coded here 4 different groups, and you look at that in 3 dimensions, and you take a look at that molecule and you look at its mirror plane, your mirror image, right? That would be the mirror image of that molecule. Is that the same or is that different? And again, if you think about superimposing those molecules, and you can see if everything lines up. And I'm just going to flip that one on the right here for you to kind of align it. I've just basically rotated it so I can align the red and the blue in the right orientation. Now if you think about this molecule sliding over and overlapping with the one on the left, right? The red ends up where the red is. The blue will end up in the same place as the blue is. But this green group is coming out towards us. That's in the same place as pink. It doesn't overlap. It's not the same. Okay, and then the pink atom in the back would be where the green atom is on the left. So in fact, those are not the same molecule. They're not the same molecule. They have the same number and kinds of atoms, right? So they must be an isomer of some kind. So they're isomers of each other. There's no cis or trans. There's no same side or opposite sides of some kind of rigid molecule. But they're different stereoisomers. There are groups that are arranged differently in 3 dimensions that can't be interconverted, and so those molecules are no longer identical. Okay, and this is something we refer to the relationship between molecules as mirror image isomers, mirror images that are not the same, or sometimes referred to as mirror images that are not superimposable. That's an aspect of molecules we refer to as chirality. These are what we would call a chiral molecule. And chiral molecules are everywhere. Of course, carbon is everywhere. And carbon is tetrahedral. And in many cases, we have carbons that have 4 different groups attached. So the molecule becomes asymmetric. Okay, so here's some terms that you should become familiar with. This concept of mirror image isomers we refer to overall as chirality, but specifically we call the relationship between 2 molecules that are mirror images of each other but different as enantiomers. Enantiomers. So this word, enantiomer, molecules that are not superimposable on their mirror image. And again, we're talking about a relationship between 2 molecules. They're enantiomers if they're stereoisomers of each other, which are related as mirror images. Okay? And molecules that have enantiomers, that have the possibility of enantiomers, are referred to as chiral molecules. If there's some symmetry in the molecule, that would be, sometimes we talk about that as being an achiral molecule. So there's no chirality. And specifically, when we're really focusing in the molecule in the position where that asymmetry stems from, that carbon that has 4 different groups attached to it, that's really where the source of it being unsymmetric lies. It's often referred to as a stereogenic carbon, or sometimes it's not quite correct terminology, but people talk about it as a chiral center, the center of chirality, or the point of chirality, but people use that term a lot. So this is some of the aspects, some of the terms that we use to describe these kinds of molecules. Okay, so molecules which are asymmetric, their mirror images are not the same. So they're not the same. These are enantiomers, like I showed on the top, enantiomers. And molecules which have some plane of symmetry in it, like difluorochloromethane, has a plane of symmetry to actually bisect the molecule. That is achiral, not chiral. Its mirror image is the same. It's the same model. So there's no possibility for having enantiomeric isomers. Okay, so it becomes important to recognize where chirality is and how we can recognize it, and how to identify it and see it. And chiral molecules are everywhere. And the molecules that make up most of the biomolecules in our bodies and in plants and everything, they're chiral. They have plenty of carbon atoms within them that have centers of asymmetry or stereogenic carbons. For example, sugars. Okay, look at this. We're going to take a close look at glucose again in a little bit, but this is a sugar molecule. I've drawn it as the chair conformation. How many stereogenic carbons are present in that molecule? Can you identify them? Can you see that there are carbons that are asymmetric? You only need one in a molecule to make it asymmetric, but you can certainly have more than one. And in fact, if you look at any of the carbons in the ring, this one, this one, this one, this one, and this one. There are 5 carbons in that ring, where if you look at each of those carbons are attached to 4 different groups. Okay, so there's actually 5 stereogenic carbons in glucose. Okay, now, last time we were talking about aromaticity and talking about DNA, right? DNA has the base pairs in the middle, which are flat. If something's flat and just p orbitals, right, there's no possibility for chirality in this sense, because there's carbons that aren't sp3, they have to be tetrahedral. However, the backbone of DNA, which we didn't talk about last time, the backbone of DNA, anybody know what that's made of? Sugars, yeah. Deoxyribose. Those are sugar molecules which are linked together, which are holding those bases. But because those sugar molecules are chiral, DNA is chiral. And DNA actually forms a helix in a particular direction, because the chirality of the sugars, the specific mirror image of the sugars, and it's just one mirror image that's used in nature, the specific isomer that's used to make the backbone of DNA also makes this larger structure of the helix in one direction versus the other. So DNA actually only turns in one direction. So if you think about it, a screw is helical because you turn right, it goes in, you turn left, it comes out. That screw is also like a helix, right? You can see all kinds of examples of things that are chiral in the macro objects and at the atomic molecular level. Same thing with proteins. Proteins are all made up of amino acids. Other than one, they all have at least one stereogenic carbon. And the amino acids that are used for proteins are nearly all one specific mirror image of the possible 2 isomers. So they're all L amino acids that are used. That term L, anybody ever heard that? L amino acid versus D amino acid? Any idea what that means? D amino acids are the unnatural ones. L amino acids are the natural ones. The ones found actually in proteins. So what does that D and L mean? Yeah, that comes from actually chemistry and when they're looking at the physical properties. And we're going to get to that, some of those physical properties that are different between enantiomers. But the L stands for levorotatory, and D stands for dextrorotatory, and it's referring to the molecule and how it rotates plane polarized light to the right or to the left. And we're going to talk about that a little bit more in a few minutes. Well, obviously, if proteins and enzymes and biomolecules, they're all made up of chiral molecules, then that manifests itself in biological properties as well, including things like how we smell and taste things. So the smell receptors that detect, when you're smelling something, you're actually detecting molecules. Molecules are binding to proteins, which are smell receptors in your mucous membranes. Those proteins that are binding to odor molecules are chiral. So sometimes those odor receptors will respond differently depending on which mirror image isomer of a molecule it's interacting with. So for example, here are some molecules that have particularly strong odors and flavors. So you can see in these molecules there's one carbon here that is a stereogenic carbon. It's a carbon with 4 different groups attached. So carvone, this is a molecule we call carvone. And there is a minus enantiomer and a plus enantiomer. That also refers to how it rotates specific kinds of light, which we'll explain in a minute. But one of these smells like spearmint. And when we smell this, it smells like spearmint. If you take the mirror image of that molecule smells like caraway seeds. And in fact, carvone is found in caraway and in spearmint. And that characteristic smell comes from the specific enantiomer that's in those plants. So caraway plants produce the plus isomer, and spearmint produces the minus isomer. Same thing, here's limonene, is almost a similar compound, but also has a stereogenic carbon. And its mirror image is different. They're enantiomers. One smells like oranges, and one smells like a little bit like lemon, but also with some turpentine smell. So they do smell significantly different if you were to smell one versus the other. I knew a company once, a chemical company that was making single isomer molecules, and they actually made some candies. And to demonstrate this, they took candies and put these mirror image enantiomers in different candies so you could actually taste the differences and experience it. It's kind of fun. They're important for smell and taste. How else might you think chiral molecules are important? Well, in drugs. When we think about designing molecules to cure diseases, in general, many strategies involve looking for that biochemical process that's responsible for a disease, finding the specific proteins or enzymes that are not doing the job properly or doing the job too well that you want to try to block, and you design small molecules to fit into it. And so here's an example of a potential drug molecule that was designed and tested. And this blue represents a protein of a particular enzyme, a protein surface. You can see, because proteins are chiral, that the surface spaces are going to be asymmetric and handed as well. For example, this molecule that's bound in there, you can see the picture of it bound in here, right? It interacts with the protein, but because this molecule has stereogenic carbons, if you were to flip or change, use the mirror image of at either of those carbon positions, the molecule wouldn't fit, like a lock and a key. Okay. Enzymes are chiral. Oftentimes we need chiral molecules to interact with it. So if, for example, you look at those two positions, if you were to take that group that's sticking to the back and take the mirror image, it would stick out and it wouldn't bind in the right pocket. This one, if this group, instead of coming here, was pointed in that direction, it would be blocked and it wouldn't fit. Okay? So being able to think about the three-dimensional structure and the particular stereoisomers that are necessary for the kind of interactions between molecules that we want is really important. Here's a real case study. This is a molecule, it's a drug called thalidomide. Anybody ever heard of thalidomide? Anybody know the story of thalidomide?
Was it used for control back in the 60s?
In the 1960s, it wasn't birth control, but it was used to tame the symptoms of morning sickness. So it was actually beneficial for morning sickness, and it had some sedative properties for that. But the problem is, when this drug was on the market, it was never on the market in the United States, but it was on the market in Germany and in Canada, and lots of birth defects started showing up from mothers who were taking this drug. A really unfortunate situation. And it turns out, because there's a stereogenic carbon in this molecule, the beneficial properties that were observed for thalidomide came from the molecule which has one particular stereoisomer at that position, one particular configuration. If you take the mirror image of that molecule, it's the mirror image that was responsible for the birth defects. Actually, this isomer on the left is perfectly safe. It doesn't cause birth defects at all and has the beneficial properties that they were looking for in this drug. So that's, that was a big learning lesson for the regulatory agencies, for the pharmaceutical companies. Since this time especially, this is one of the biggest problems that had come out of this. By the way, not all chiral drugs have a bad isomer. Sometimes it doesn't matter. They could both be good, or one just doesn't do anything. But because of this, it's been mandated now that any drug that's developed that has chirality, that's a chiral drug, has to at least be prepared separately, purified separately, and tested separately to know the actual properties of each isomer. And it can't be sold as a mixture of isomers unless that's been proven that both isomers are safe. So this has created quite a challenge for synthetic chemists, because it is very, very difficult to synthesize a single stereoisomer and purify them. Okay, well, the story of how we discovered and recognized chiral molecules is kind of interesting, and I want to spend a little time going through this story. Enantiomers are interesting because they have a variety of physical properties. Well, molecules have physical properties that we know and recognize, right? They can have a certain temperature that it melts, a certain temperature it boils. They can have different properties with various spectroscopic techniques. They can have various properties with density and things like that. But it turns out enantiomers, mirror image isomers, have all those physical properties are identical. There's no way to tell them apart based on those physical properties. The only difference that we can find, other than how it interacts with other chiral molecules or chiral spaces, is that they change the direction of plane polarized light in opposite directions. So that's the one physical property interaction that occurs that's different between enantiomers. And this was discovered before we knew much about molecules and structure and didn't know anything about tetrahedral carbons. These properties were discovered. The ability to actually make plane polarized light goes back all the way to the 17th century, where if you take a polarizing filter, just like you have in your sunglasses, light travels in waves, and if you look at the direction of light, those waves are in all directions. They're vibrating in all directions along the path of the light traveling. And if you take that light that has those waves in all directions and pass it through a filter, a grating filter, which has slits, very narrow slits in one direction, it blocks all of the light waves except the ones in one direction. Okay, so this is what we refer to as the light being polarized into one plane. So what comes out the other end is what we refer to as plane polarized light. And this was discovered quite a long time ago, this light. So when chemists started taking molecules without really understanding what we know today of structure and the atoms, and testing it with all kinds of different tests. They boil it, they melt it, they burn it, they do this, they do that. They looked at how light passes through it as well. And they discovered that chiral molecules, certain molecules will bend the plane of that light as it travels through a solution of it. Okay, and this was discovered in the 18th century. Actually, it was close to the 19th century, 1815. He discovered plane polarized light would be rotated, the plane would be rotated with certain molecules, certain organic molecules. So again, here's kind of a schematic of what we see for this property. So I talked about the L and D amino acids. That means the light is bent to the left, or that plane is bent to the right. So again, we generate plane polarized light here, which is all the wave traveling in one direction. As that light passes through a sample, a solution containing chiral molecules, the plane of that light is tilted, one direction or the other. So they can observe, and you can see just by taking another polarizer and seeing at what angle the light comes through it, you can see at what direction that plane is. So they observe that light will be bent in one direction. And now we know that if we have the mirror image isomer, it'll be bent the opposite direction, the exact same amount. So this is a physical property, the only physical property that's different between enantiomers. But something that we can measure, and that's great. So this is what we refer to as optical activity. Molecules, you might hear that chiral molecules are optically active. That means they bend plane polarized light. Molecules with symmetry won't bend the light. Only chiral molecules. And chiral molecules that are pure, or not 50-50 mixtures. Here's an interesting molecule, tartaric acid. That's the structure shown here down on the right. Tartaric acid. Tartaric acid was probably the molecule that people really studied early on to determine and find out about stereochemistry. And quite not by intent, they learned this just by observation. So you'll notice tartaric acid in there. There are two carbons that I've highlighted with those green stars. Those two carbons are stereogenic. They're asymmetric. They have two different isomers. Mirror images, if you take the OH on the left one could be up or down, and the OH on the right one could be up or down. This molecule was, of course, a really important molecule at the time because it was obtained from a natural source, actually from wine. It crystallizes out from wine because grapes form a lot of tartaric acid. And so all of these crystals that are formed inside the barrels or on the corks, these are all various salts of tartaric acid. And Louis Pasteur, you might have heard of him, not only did he develop methods of pasteurization, but he also was studying tartaric acid. He was a chemist. And he was the first person to separate mirror image molecules. Again, if there are no physical property differences other than light, how do you actually physically separate them? Well, it turns out that how they crystallize, sometimes chiral molecules crystallize in specific ways. So he noticed that the crystal forms, let me back up. The crystal forms were of mirror images. On the macro scale, he could actually physically see this. So this particular salt, sodium ammonium tartrate, formed really nice crystals. This is a picture actually of some of Louis Pasteur's samples, actual samples of tartrates. This is on display at a university in Paris. And I don't know if you can see, you probably can't read that. There's some racemate of potassium and sodium tartrates and all these different tartrate samples that he has. It's very interesting. But here's what he said. Here's what he found. I'm quoting some of the things that he reported in his early papers in this area. He found crystals of tartrates that were hemihedral in shape. And he noticed that some of those crystals incline to the right and some incline to the left. You can see a drawing of those crystals here. And he took a magnifying glass and a pair of tweezers, and he physically separated those crystals that were shaped to the right and those that were shaped to the left, and separated them into separate piles. And when he did that, he saw that the crystals deviated the plane polarized light in one direction or the other. So before this time, many molecules in nature are chiral and as only one form. Tartrates happen to be a mixture, and he was able to separate both of them, the left-handed and the right-handed tartrates. And there he could directly observe for the first time that, yes, mirror image salts of the same molecule, or mirror image crystals, rotate that plane polarized light in different directions. So it was pretty profound discovery. And here's where our DNL come from. Dextrorotatory refers to rotating that light to the right, and levorotatory refers to rotating that light to the left, from the viewer of observing the light, I presume. Okay, interesting, huh? It's kind of a fun story. So Louis Pasteur did a lot of things, not just in the biomedical field. So, actually, with a little more detail, what we know now is that how much that rotation occurs depends on how many molecules the light hits before it comes out the other end. So each molecule will tip it a little bit more. So in order to come up with a way to standardize this, the rotation depends on how many molecules. The light, the instruments are actually looking at a specific wavelength of light called the sodium D liner, 580 nanometers. That's just what the instruments measure these days. And then they simply standardize it based on the length of the cell, how long the light has to pass through, and the concentration, because that then factors out the number of molecules it actually hits. If you change the concentration or change the length, you're going to hit more or less molecules, right, as the light goes through. So we have a standard rotation number, and so any chiral molecule now has a standardized number for it. They all rotate light a little differently, but referred to as this alpha. The D stands for sodium. The alpha rotation number, standard rotation. And this is now tables and tables of molecules that we know what the actual optical rotation standard number is. Okay, so we can compare them. Okay, so it all comes down to those molecules that are chiral interact with plane polarized light. One of the observations that Louis Pasteur did, he talks about it here. When I took an equal weight of each of the two kinds of crystals, the left and the right, took an equal amount and mixed them together, he says they were indifferent towards the light and neutralized the equal and opposite deviation. So in essence, he found that if you mix them together, the two mirror images together in equal amounts, then the light is no longer deviated. Okay? And that makes sense. If you have a 50-50 mixture, one will rotate the light a little bit to the right, it hits the next molecule, will rotate that light a little bit to the left. So if you have an equal number of both, there's going to be no change as the light comes out the end, right? Everything is going to cancel. And actually, if we have something other than 50-50 mixture, we'll see partial optical rotation in one direction or the other. That's another term for you, racemic mixture. A racemate or a racemic mixture is a 50-50 mixture of enantiomers. So tartaric acid normally in grapes is a 50-50 mixture of enantiomers. and Louis Pasteur separated them physically because they crystallize differently, fortunately. And then if you mix them 50-50 together, the right-handed light rotation is canceled out by the left-handed right rotation, and we don't see the optical activity. So sometimes there are molecules which are chiral, but are present in equal amounts, the two isomers, that we can't tell because they're 50-50 mixtures, or racemates. Okay, interesting, huh? I hope you find it interesting. Well, the story of at the time that Louis Pasteur was doing this, they didn't know what the structure of carbon was. They didn't know atomic structure nearly like we do today. There was even debate about whether carbons were tetrahedral or not. Nobody really knew. And it was in 1874 when this observation that they see about chiral molecules was related to what might be the structure of a tetrahedral carbon. And so one of the main players in this area was Van Hoff. Van Hoff proposed that carbon that has 4 groups attached was in this tetrahedral shape that we know today. And that if you have 4 different groups, then you could have those molecules exist as mirror images. So it was only not that long ago compared to some of the earlier discoveries, 1874, when they were thinking about tetrahedral carbons in 3-dimensional space. The story's kind of interesting. When Van Hoff proposed that, he wrote this up, these ideas of his about tetrahedral carbon and asymmetry, wrote it up and published it back then in science they would publish pamphlets or publish papers, publish letters to journal editors. And he was a very young chemist. Young guy, just starting out his career. At the same time, there was a chemist named Kolbe. And he read this report from Van Hoff and said, "Oh, this stuff is crazy. What is this guy dreaming of? It can't be that." He was, and I think it was a little narrow-minded, but he really criticized, a scathing criticism of Van Hoff's work. And I want to share with you some of this because I think we as scientists don't write like this anymore. And it kind of tells you that even within science, scientists can be closed-minded and criticize people, especially people who aren't very well known yet. So he's talking about the work of Van Hoff and lamenting about how the state of education of chemistry is getting really, really bad. Of course, you all hear lots of professors talking about students not getting it, right? So he says, he published an article recently about the decline of chemical research and the lack of well-rounded and thorough chemical education, okay? And he talks about complaining about chemistry professors doing disservice to the science because they're not teaching right. So they're coming up with all these crazy ideas. As a consequence, there's an overgrowth of the weed of the seemingly learned and ingenious. The seemingly learned and ingenious, but in reality trivial and stupefying natural philosophy. And he's getting to the point where he's going to talk about Van Hoff. This natural philosophy has put aside exact science and is being dragged out by pseudoscientists. I love this. Pseudoscientists from the junk room, which harbors such failings of the human mind, and is dressed up in modern fashion and rouged freshly like a whore, who one tries to smuggle into good society where she does not belong. And you don't find that in scientific papers today. Although it would be kind of fun. So essentially what he's saying is, and again, he's complaining about Van Hoff, and he's saying, well, they're presenting these terrible ideas that shouldn't be brought to good company because they're just idiotic, and dressing them up and making them look nice. That's what he's saying. Whoever considers the apprehension to be exaggerated should read, if he can manage it, a recently published pamphlet by Mr. Van Hoff and Herman, his collaborator. So essentially he's saying that this work by Van Hoff is garbage. Teams with fantastic triples. I would ignore this paper as so many others if it were not for a renowned chemist. Another big name chemist was actually supporting Van Hoff's ideas. And he complains about another famous chemist who was protecting the nonsense. That's kind of interesting. He goes on, some interesting words here too. Van Hoff, and he talks about, he's complaining that Van Hoff is just a young guy at a veterinary school. Van Hoff is employed at a veterinary school, appears to find exact chemical research not to his taste. He's saying he doesn't know what he's talking about. He deems it more convenient to mount Pegasus, evidently loaned from the veterinary school, to proclaim in his article how to him on the chemical Parnassus, which he ascended in his daring flight, the atoms appear to be arranged in the universe. So he's saying Van Hoff's head's way up in the cloud, and he's coming up with all these fanciful ideas about how the structure of the carbon is tetrahedral. He doesn't believe it. And he goes on to talk about more details of what Van Hoff was trying to propose. And just goes on to talk about how Van Hoff is crazy. But without actually really looking at the evidence and the science. And for the record, Van Hoff received the first Nobel Prize in chemistry for his work. So Van Hoff actually was correct. And it's those insightful ideas that people come up with that go against conventional thought, I think, that often bring about changes in how we think about things. And it turns out that the tetrahedral carbon was spot on. We know a lot now, a lot more about the structure and three-dimensional arrangements of atoms. Okay, so we have this issue, right? We have molecules that can be asymmetric and present to us a new kind of stereoisomers, and we need to have ways to name them and note the difference. And you've seen some of the names on some of the molecules maybe I've shown before, some of the older systems where we use D or L, but that's referring to the direction of light and doesn't say specifically what configuration it is. And it's more a description of the physical property. But what we have come up with is a very systematic way to name the molecules, which you can see here. And you can see these molecules, this 2-chlorobutane. These molecules are named based on the specific configuration of that stereogenic carbon. And you'll notice in one case, the molecule has an S in front of it, and the other one has an R in front of it. Those are the two enantiomers. And that S stands for left, or R for right. Left from the word sinister. Right from the word rectus, Latin. So how do we come up with that? How do we know? What do we mean by that's the left molecule or the right molecule? Or specifically, this is referring to the configuration at the stereogenic carbon that we're talking about. So we have 4 groups. We just need to come up with a systematic way to differentiate 4 groups and then talk about whether the arrangement goes in a certain direction. So I want to go through this with you. The way we prioritize groups we've seen before, it's exactly the same as how we prioritized groups for determining E and Z on a double bond which has 4 different groups on it. Okay, so CIP stands for the Kahn-Ingold-Prelog rules. Do you remember we talked about assigning priorities to groups based on atomic number? And then, of course, we need to go out layer by layer if we can't make a distinction right away. So that's what we've done here. We have 4 groups on carbon, and we assign the priorities from 1 to 4. So if you look at the specific carbon that 2-chlorobutane, that the chlorine is attached to, that particular stereogenic carbon, there's a chlorine group, there's a hydrogen, there's a CH3 methyl, and then there's a CH2CH3 ethyl. Okay, and so hopefully it's easy for you to see that the priorities would be one, chlorine would get the highest priority, right? If there's a hydrogen, hydrogen is always the lowest priority because everything else is going to be carbon or something else. So chlorine is 1, hydrogen is 4, and then we have CH3 versus CH2CH3. And if you go out, carbons, the first atom is a carbon, the next atom is all hydrogens on one and another carbon on the ethyl. So that's why this group gets second priority and then methyl third priority. So what we do then to think about whether it's S or R is we have to view the molecule with the lowest priority group pointing away from us. And I've drawn it that way. It's not always drawn that way, so sometimes this takes some gymnastics of those, thinking about that in your head to see and orient those properly. But here the hydrogen is pointing to the back. That's the lowest priority group. And then you look at the remaining 3 groups, and if you go from 1 to 2 to 3 in a counterclockwise direction or to the left, it's the S isomer. And if you go from 1 to 2 to 3 in a clockwise direction or to the right, it's the R isomer. And it's that simple. And I say simple because what's not simple is making sure you're looking at the molecule from the right orientation to put the lowest group towards the back. Okay, what if I drew it, so here's the thing, if I drew it like this, let me undo that. If I drew it like that, we have priority 1, 2, 3. Which direction is it going? Is that R or S? It's R. Okay, why is it R? Well, the lowest priority group, the hydrogen here, is pointing out towards us. So you actually have to just flip it around. And if you flip it around and look from the other side or change it, and then it's hard to see sometimes in your head, but you would see that then if you're looking at it from the right orientation, then you go clockwise from 1 to 2 to 3. Okay? Now, one easy thing with this is if the lowest priority group, in this case hydrogen, is pointing up, you can look at what's there 1 to 2 to 3 and take the opposite of that direction because you'd be looking at it from the back. And it's easy to see that. Well, what if I drew it this way? What if I put the lowest priority group as one of the bonds in the plane of the board? Can you also rotate it and look at it from the right direction? Or see which way it goes? What's the configuration of that molecule? Anybody?
R.
It is not R. It is S. See, it can be tricky. It can be tricky. Why is it S? Well, let me rotate it so the hydrogen is pointing to the back. So I'll just rotate it just a little bit. And so I would have this, and that hydrogen goes towards the back. This group comes out, swings out towards us. 1, 2, 3, it would be counterclockwise, it would be yes. But it's hard to see that, right? It's hard to see that. That's why we've spent a lot of time really trying to think about envisioning three-dimensional structure, because it's really important here to be able to see the molecule right. Now, there is a trick that you can use that will help regardless of where the hydrogen is pointed. And that's to use your hands. And it's a pretty good trick because all you have to do is point however the molecule is drawn, point your thumb in the direction of the lowest priority group. And the direction your hand curls is the direction the rest of them go 1 to 2 to 3. So in this molecule on the top, with the hydrogen going to the back, I need to point my thumb to the back. And it's going that way. If I use my right hand to do that, it doesn't match, right? So I need my left hand to do that. See how I'm doing that? So with our structure on the right, I need my right hand to curve 1 to 2 to 3. Your left hand won't curve in the right direction. If it's drawn like this, we can do the same thing. Okay? Now we just point our thumb in that direction, so does it go chlorine to methyl this direction? Or if I do it this way, it's going chlorine to CH3 to CH2. It doesn't work with my right hand. I know it's going to take a lot of practice to get this, but I think given enough examples, you should be able to get this down. And I put this in the slides, D being the lowest priority group, A being the highest in this particular figure. You point the D behind towards the back, and then it's whichever hand curls in the same direction of the priorities. That's the left or right, S or R. Okay? Okay, let's look at some more examples. Here's a molecule called sphingosine. Sphingosine is an interesting molecule that is important for docking things to the surface of cells because this long hydrocarbon chain that goes into the cell membrane bilayer. And then those groups on the top, this oxygen, those two OH groups and nitrogen, they attach onto other sugar molecules and append on different groups onto the surface of cells. So this is an important molecule in biology. But if we look at this, there are two stereogenic centers. And again, what you're looking for is carbons with 4 different groups on it. And remember, hydrogens aren't written usually in our line structure, so you have to know where those hydrogens are. So those there's two configurations there. Let me just blow that up, and we'll take a close look at that. So if we look at the first carbon that has an asymmetric carbon, that's this one. Okay. Notice that carbon is not a stereogenic carbon because what's on there? 2 hydrogens, so 2 groups that are the same. We have to have 4 different groups. So if we look at this carbon, the hydrogen that's not drawn there, I want to first make sure I know where that is. So that hydrogen has to be coming out of the board towards us to make that tetrahedral carbon. Right? So then we know where the hydrogen is. We know that's the 4th priority. Now we have to look at nitrogen, carbon, and carbon. Which of those groups is the highest priority? Nitrogen. So that's the highest priority group right there, nitrogen. Now we have to see what's 2 and what's 3. So this, and again, this is the same rules that we used when we looked at EZ isomers. We use the same priority identifying rules. So this is a carbon attached to 2 hydrogens. So 2 hydrogens and an oxygen. This is a carbon attached to 1 hydrogen, 1 oxygen, and another carbon. Okay, so right there we can make a distinction. Which has higher priority? The group on the right. So this direction, that's priority 2, and that's priority 3. Okay, I'm going to clean this up and put those priorities back on here. So hydrogen is coming out towards us. Hydrogen is 1. This is 2. This is 3. Sorry, nitrogen is 1. hydrogen is 4. Okay? So the hydrogen is the lowest priority group. It's pointing out towards us, and we need to look at the direction if that's pointing away from us. So either you can envision looking at this from behind, or turning this molecule 180 degrees in your mind, or you can point your thumb out and see which way your hand curls to go 1 to 2 to 3. That's probably the easiest way. So which hand do you need? I don't see anyone with their hands up. Try again. So if I use my right hand and point my thumb this way, my hand's curling that direction. Nitrogen, so 1 to 3 to 2. I need this hand pointed out, and you go 1 to 2 to 3 in that direction, and that's the right, that's the correct, that's the one that fits. So left hand, the configuration at that carbon must be S. Okay, it's the S configuration. But this isn't not just one. You have a question?
Yeah, I was just confused about the hand.
Yep.
Depending on what hand you use is how you can tell S or R.
Yep. Because only one hand works.
That makes sense.
That's why it's a good trick to use. Okay, so that configuration is S, but there's a second configuration. So this molecule actually has 2 stereogenic centers, and we need to identify the configuration for both. Because this molecule could be SS, it could be SR, it could be RR. Those are all different. Or RS. There are 4 different possibilities, right, of combinations. So let's take a look at this configuration. This one might be a little bit easier to see. We have a hydrogen, oops, not coming up. We have a hydrogen going to the back. The highest priority group is oxygen. I hope you recognize that. Now we have a carbon here and a carbon here. We can't distinguish, so we need to go out to the next layer. So the carbon on the right is attached to what?
Another carbon.
So this carbon is attached to another carbon. It's attached to another carbon. If we think about the double bond, it's attached to another carbon and a hydrogen. That's a carbon. Okay? Carbon, carbon, hydrogen. This carbon is attached to carbon, hydrogen, and nitrogen. At this point, we can say that this group gets the higher priority. Okay? Because of that nitrogen. So if I were to look at the again, I'm focusing, keep in mind, I'm focusing just on this particular stereogenic center now. On this specific stereogenic center, oxygen is 1, this direction to the left is 2, this direction to the right is 3, and the hydrogen going to the back is 4. Okay, for that center. Okay, we already did this one, we don't care, we already know that sense. Now we have to look at this center. 1, 2, 3, 4. And which hand?
Right.
You need your right hand. Like that. That's right. So it's R. Left hand doesn't work. Or you can see that this is going, hydrogen's pointing in the back. It's easy to see it's going clockwise. Okay. So the configuration for this molecule, did I have it on there? No, I didn't. Configuration for this molecule would be a specific isomer where this configuration, I'll get this one of these days. Come back. Okay. Where this configuration is the S configuration and this configuration is the R configuration. And when we name this, and I'm not going to give you the whole name for this molecule, but when you name a molecule, you need to name the configurations at the centers they are. So the name for this would begin, let's see, carbon 1, 2, 3, 4, 5, etc. The name for this molecule would begin with a 2S, 3R, maybe, I don't know if the parentheses are there, 2S, 3R, and then the rest of the name. To note that The number 2 carbon is a stereogenic carbon and it has S configuration. The number 3 carbon is a stereogenic carbon, has the R configuration. Okay, so that's put into the name. Okay, here's some amino acids. Amino acids, they make up proteins, but they all have stereogenic carbon. 2 of them here, one is alanine, one proline. So what is the configurations for these carbons? Figure them out. Where do you start? Right? Identify the stereogenic carbon. Make sure you know where the hydrogen is, if there's hydrogens on it. So in this case, we have 2 bonds that are in the plane of the board. Here, we have the CH3 going to the back. The hydrogen must be coming out towards us. By the way, I see people draw all kinds of things. Notice that when I draw line structures and the connection between 2 bonds is pointing down, the other 2 bonds are away and towards us, pointing away from that point. So that would be, I could draw the hydrogen there. That would be the same actually as if I drew the hydrogen here. As long as it's going toward down and out from that point. Okay. What I don't want to see, and which will really confuse you and mess things up, is if you were to do this. That looks just so wrong because it completely misrepresents the tetrahedral structure and is meaningless. So don't do that. So you can point it down. I'll just put it here again. But coming out of the board towards us. Okay, so now assign priorities. What's number 1? Nitrogen. What's number 2? The carbon attached to oxygens. Number 3 would be the methyl, and the hydrogen is 4. How many say it's S? 1 in the back, 2, 3, 4, 5, 6, 7, 8. Hopefully pretty soon everyone will be raising their hand and saying it's S because it's S. Why is it S? Well, notice to go from 1 to 2 to 3, the way it's drawn on the board, I have to go clockwise, but remember the hydrogen needs to point to the back, so you need to turn it around and look at it from the other direction. To go from 1 to 2 to 3, you're going clockwise, counterclockwise. Or, easier, you need your left hand pointed out to get the direction from nitrogen to number 2 to number 3. Your right hand would go the wrong direction. Okay. So that is S. So this is S-alanine. I don't need to put a number because there's only 1 stereogenic carbon, so it's obvious where it is. S-alanine. Or L-alanine, we'll see it listed because L-alanine is the natural one. Happens to rotate the light to the left. Yes?
What happens if you do put the hydrogen on the other side?
Well, let's look at that. Good question. What happens if I put the hydrogen on the other side? You mean over here?
Yep.
Okay. As long as it's pointing down from that point of the 2 bonds, in the right place, it should be exactly the same because the lowest priority group we don't really care about. It's the arrangement of the other 3 groups, and we haven't moved those. Okay, it's still going. We haven't changed the orientations of anything else. So it's still. This is still 1. This is still 2. This is still 3, and hydrogen is still coming out, so we still need our left hand to do that. So it doesn't matter. That doesn't matter. What may matter is if you put things up here where it's wrong. That's where sometimes, depending on the structure, it can get bad. So don't do that. Okay, so that's S. And proline. So notice I'm putting that towards the back. Here's the two bonds in the plane of the board, and I'm going away from that point, either there or down here, it wouldn't matter. And priorities. N is 1, carbon with oxygen's 2, the carbon, the CH2 up there is number 3. And so at that configuration, hydrogen pointing to the back. Left hand pointing to the back to get to it would be S. Okay, making more sense now? Okay, I'm going to stop there today. We'll have exam on Tuesday, and then we'll come and revisit this and talk more about stereochemistry on next week, Thursday.